What we need to talk about now is DNA replication. To replicate means to make copies of yourself or copies of itself. So when you were born, you were only made of one cell. And now you are trillions of cells. And all of these cells have DNA in them. So there has to be a way for DNA to make copies of itself so that little bits of DNA or all the DNA that you need are in each and every cell that you have made. So before a new cell is made, DNA goes through this process of replication. How does this work? First thing, the twisted double helix has to unzip. So it unzips like this, or unwinds, excuse me, I should say unwind. So we have the, it's not twisted anymore, it's laid out flat. Then I'm going to refer to what's on the screen, on the refrigerator here. So on the refrigerator, I just put a little chunk of DNA up here. I would use these in class with you. They're magnetic. And I use different colors for the different bases. So my greens here, or yellowish green, are going to be my thymines. My blues are my adenines. My orange is guanine. And my pink is cytosine. So this is a little chunk of DNA that has unwound itself, so it's laying out flat. First thing that happens is this little molecule here called an enzyme comes along and it unzips, it breaks the bonds between the nucleotides and it unzips the DNA. So the DNA unzips and it spreads out kind of like this. All right? And it'll unzip all the way up, like a big zipper being uncoiled. Now, the next thing that's going to happen is new nucleotides that are floating around in the cell they're going to go out and grab them, and they're going to bring them back to the DNA here to make the new copies. This side and this side of the DNA are kind of like the template or the model that it's going to use so it can build the new strand. So if you look, this guy right here is a T. What we need to match with him is an adenine. He'll come in here and fill in. Cytosine, here's a guanine. That'll fill in. I'm going to move this out a little bit further so I can get in here. Guanine will pair with cytosine, and it just keeps reading the code. It does the same thing to the other side here. So adenine will go find a thymine, fill it in. Guanine will go find a cytosine, fill it in. And this cytosine will find a guanine. And it keeps working its way on up and filling in and filling in. So it unzips, it fills in the complementary bases by reading this side and this side. So if you look at it, this side of the strand right here is part old. This is part of that's new. This is the part from the old. This is the newer strand. So when we get done, we're going to have two strands of DNA when we used to have one strand, and then it'll coil itself back up again in the double helix formation. So it's kind of like half old and half new. And this happens all the time in your body. It's happening right now, and you don't even know it. You're making new DNA for new cells. Cells don't live forever. Some cells last longer, brain cells last longer than skin cells do, and so on and so forth. But you've got to make new DNA. Now, in this process of replication, sometimes, unfortunately, there are mistakes made. And these are called genetic mutations. And there are three kinds of genetic mutations. Fortunately for us, our bodies have a check system that reads the DNA when it's done copying itself and looks for errors. And if it finds errors, it usually corrects them. It's when it doesn't correct the errors that we have problems. Sometimes genetic mutations turn out to be favorable, and they give you a certain leg up on the competition, give you an advantage. Like perhaps a long time ago, ducks didn't have webbed feet. There was some kind of genetic mutation that caused a duck to have slightly webbed feet. Hey, that ended up being a good thing, and it got enhanced and enhanced over time. So some scientists believe that genetic mutations are like the driving force of evolution. Sometimes they're bad. Cancer is probably the number one thing you think of when you think of genetic mutations. And every time when I talk about this, everybody's always like, oh, it's like the X-Men. No, that's a little different. That's a cartoon, all right? So here we go. Let's just say this is my original strand. I mean, DNA is much bigger than this, much longer, but I'm just doing a little chunk here. So I've got C, G, A, T, G, C, and I have to have the complementary side here. So C goes with G, G goes with C, A with T, T with A, G, C, G, or CG, excuse me. That's my little strand here. All right, so let's say this happens. When it copies itself, one of the new strands looks like this. Now, this strand is different than this strand. There's something wrong with it. What you have to do is you have to look at these two and say, okay, what's wrong? And you go through and you say, okay, well, this is C, that's C. This is T, that was G. Uh-oh, that's not right. This is wrong. Let's go to the next one. A, that's right. T, T, that's right. G, G, that's right. C and C, that's right. Everything about this is right except for this pair right here. This is an AT pair. It should have been a GC pair. Now, most of the time, your body will read that code and it'll say, this is wrong. It'll cut out this bad part here and replace it. And it does that all the time. It's supposed to do that. So when we get older, that process kind of breaks down, like makes you more susceptible to having problems. This is called a substitution. And substitutions are generally not that big of a deal. Mutations are all not great. But in terms of the three kinds, this one's not as bad. This would be like if the whole class was sitting at the football game, we're in the bleachers are packed, we're side by side, Billy gets up, leaves, and Jenny takes his place. That doesn't really affect the rest of us because one person got up, one person sat down, it didn't really affect anybody else. Now, another situation here. Look at this strand and compare this strand to this one. C with C, G, G, C, it's supposed to be A. But here's our AT here, here's our TA here. Something's wrong. 1, 2, 3, 4, 5, 6 pairs. 1, 2, 3, 4, 5, 6, 7 pairs. The problem here is this guy is not supposed to be there. He's an extra pair. So this is called an insertion or an addition mutation. You can call it either way. Some people say insertion, some people say addition. Means the same thing. You got an extra pair in there that shouldn't be. This would be like, all right, we're at the football game, bleachers are packed. Instead of Billy getting up, somebody else comes, Mason comes, and says, hey guys, can you make room? And he squeezes in and everybody has to shift down and everybody kind of gets crammed together. So an insertion mutation is not good because it shifts the whole entire code. And we'll learn about reading this code later, but it shifts the entire code down. I'm going to try to show you an example of that here in a second about shifting the code. The last one here is called a deletion mutation because check it out. 1, 2, 3, 4, 5 pairs. It's supposed to have 1, 2, 3, 4, 5, 6. So it's missing something. So let's check. CG, that's fine. Uh-oh, this pair right here should be right there. It's missing that pair. This is gone. So that has been deleted. That would be like if Billy got up, went to go get something from the snack bar, never came back. The rest of us could all spread out and shift because, hey, we got more room now. So it shifts the code. So I got an example here that I've seen used before that I think might be helpful. All right, let's say we want, let's say we want our code to be like, let's use the letters of the alphabet instead of ATCG. So we have let's go Western. That's what it should say. If we look at this statement, it says, after it replicates itself, it says let's mow Western. Well, what the heck happened here? That's fine, that's fine. Uh-oh, this G has been replaced by an M. So this would be like a substitution mutation. Let's mow Western. Can you still kind of get the gist of what's going on when you read this sentence? I would say yes. You could probably figure out what that's supposed to be. So that was only a slight error. I like to think of it as if you were taking a Scantron test and for number 5 you put the wrong answer, then it only affects number 5. It doesn't affect the rest of it. Okay, let's look at this example. So it copied this statement, but then it says lesg ow estern. Man, that's really messed up. Let's see what's wrong with it. Well, I know this T is gone. Used to be right here, it's gone. And because it's gone, everything shifted down. That's a deletion, and everything shifted. So now the whole code is jacked up and it doesn't look right at all. That's not a good thing. Deletion mutations are bad. So it messes up everything. That would be like if you're taking a Scantron test, you simply forgot to answer number 5. You totally skipped it. You deleted it. And for number 5, you put the answer for number 6. And for number 6, you put the answer for number 7, and so on. And you messed up the whole thing. I don't know if anybody have ever done that on Scantron where you skipped one and you messed up everything after it. I did it once in college, and I missed a whole bunch in a row. Okay, next. Let's see. What's wrong with this guy? So, let me see. L-E-F-T-F. Where did F come from? So that has been inserted or added. So that has changed the code a lot. So that's like left sk ow estern. So that's really messed up. So the one mutation that's not that bad, I mean, they're all not, you don't want to have any mutation, but the one that's not as serious as this substitution mutation. You only have one little thing wrong. Deletion shifts the code, insertion shifts the code. Oh, and one more thing. I also will talk about things called mutagens. Mutagens are things, sometimes mutations are just random, they just happen. But sometimes there's things that you can put into your body that would increase your odds of mutations, genetic mutations. Those would be called mutagens, or I don't know if you ever heard the word carcinogens. Carcinogens would be like cancer-causing substances like tobacco, trying to think here. There's a chemical called asbestos, ultraviolet light, too much of it, you get skin cancer, you're out in the sun baking yourself all day. Certain chemicals can be carcinogenic, so those are things that you kind of want to protect yourself against. Put sunscreen on, don't smoke, don't chew tobacco, don't put yourself at risk. Okay? All right.